Hello everybody! I hope you can hear me. It's a bit windy out here on Carrie Bow Cay where we're coming at you live. So we're here live on Carrie Bow Cay, a Smithsonian field station, 15 miles off the coast of Belize. Pretty remote. I'm going to turn you around and say hi. Hi everybody. So my name is Emily Frost, and I manage the Smithsonian Ocean Portal website out of the Natural History Museum in Washington, D.C. is where I'm normally based. But you can see I'm here on this island, which is a Smithsonian field station, 15 miles off the coast of Belize. And we're here with MarineGEO researchers who are looking at the coastal ecosystem diversity, and we're going to get some time to talk to them today in person. They've taken some time out of their research to give us some updates on what is happening. So yeah, as I said, we're 15 miles off the coast of Belize at Carrie Bow Cay Marine Station. Please ask questions, ask us any questions you want. I'll try and answer them. If I don't answer them now, I will get to them later, and let's go meet Scott. I'm going to give you the good view again. So we're going to walk around. We're standing with me on the top porch of the main house on this island. It is an island that is less than an acre in size. It's quite small. You can see it's pretty windy, and we're going to wrap around here on the porch and go to some place where you can hear a little bit better. Let's keep moving. So we're gonna, as I said, we're gonna talk to Scott Jones, who should be just right around the corner waiting for us. Oh, look, there he is. He is a program coordinator here at Carrie Bow Cay, and you can see Zach too in the background. There's only actually 7 researchers max at a time on the island. Is that right?
That's correct.
Cool.
Yep.
So Scott, I promised readers of the Ocean Portal to get to the hard-hitting stuff first.
Okay.
I want to know what the situation with the bathrooms here is.
Oh, the bathrooms. Yeah, this is actually a pretty common question. So when you have 7 people on a small island, one of the things you have to account for is how to deal with human waste, of course, and we want to have this little impact as possible on the reef. So what we do is we use composting toilets, which kind of sounds scary at first, but they actually work really well. It's a regular commode toilet seat on top of a drum, a rotating drum that holds waste and peat and microbial mix, and it gets aerated every day by the station manager, and they work pretty well in keeping the waste out of the water. Yeah.
Great.
Yeah.
I have had to use them for the past week, and
Yeah.
they are great.
Cool.
Right. So, can you tell me how long has the Smithsonian been doing research on Carrie Bow?
Yeah, a pretty long time. So this is our 45th year of operation. So in 1972 is when the Smithsonian scientists first set foot on the island, and immediately, I think, identified it as a great spot for a research station, and it's been going ever since.
Great. And why did they think it was such a great spot? What makes it kind of special here?
Well, the researchers at the time were looking for a permanent base of operations for really intensive investigations into tropical marine environments like this. And Belize is a very unique reef system. It's a barrier reef, and there's not many places like it in the world where you can see so many different types of reef formations and reef types and different types of organisms interacting with each other.
Great. And so you said that they how did we came across this island, and how does that, do we own the island, or
No.
what's the history of that?
So the history is, the scientist, his name is Calcification. He's worked at the Museum of Natural History for many years, still works there. And they were out doing research on some nearby reefs, and were heading home and missed the cut in the reef. So if you get from the ocean side to the end, you have to find a way through the barrier reef. And we're on one of the cuts, and there's one north of us. They thought they were going there. They wound up here, and Klaus said, what's that island? And Yep, so the folks that owned it at the time still own it today, and we've had a productive, fruitful relationship with that family that entire time. So I think we're on second or third generation.
Yeah. Yeah, so the Smithsonian has been doing research here for 45 years.
Yep.
And what kind of research? I know a lot goes on here and it changes frequently.
It does.
So can you tell us a bit about the kind of research?
Sure, and in a given year you can see pretty much across the spectrum of different types of marine research that people would do on a coral reef. We're a facility of the Natural History Museum, and so natural history museums tend to be really focused on biodiversity, and so of course this was a place where folks came to study and try to document the biodiversity on the reefs here for many, many years, and I'd say that still continues to this day. But now you see a lot more work going on that looks at core processes of coral reef ecosystems and how they function, and how human beings are impacting them, and that's what you're probably going to end up talking to other people today about too. So it's a great example of the kind of work that's more common now.
And finally, what is your favorite part about working here on this special place, special island?
It's sometimes I feel like the luckiest guy in the world to be able to work underwater, see coral reefs up close and personal. See the same sites over and over again and get to be familiar with them and the animals that are there. But sort of at the end of the day, when things that really stick with me are the history of this place and the people I get to work with as a part of a team, working towards one goal over the course of several days to get as much work done as possible, gather as much information as possible, and overcoming the obstacles that pop up along the way.
Great. Well, it was nice chatting with you, Scott. Get back to work.
Okay.
And for those of us, I'm going to turn us around so we can see the beautiful view. I just wanted to reiterate for those who have just joined us, we're at Carrie Bow Cay, which is the Smithsonian Marine Station, 15 miles off the coast of Belize, and it's less than an acre in size. It's pretty small. So we're going to move over here and talk to Ross Whippo, who is a biologist and the lead technician for the Smithsonian program MarineGEO. So we're going to move over here, and there's Ross. So we are standing over the station's marine lab, and you can kind of see it. I'm going to move in here. So there's lots of samples down there. Right now we've got lots of algae and seagrass ready to go, and Ross is going to tell us a bit about what the research is. So tell us, what is MarineGEO for those of us who have never heard that word or name before?
Yes, well, first of all, hi out there. It's great to be here. I hope you're enjoying
I
it.
just turned us around. Hold on one second. There's Ross again.
Live feed. Hope you guys are enjoying your tour of Carrie Bow Cay. This is my 4th time here, and it's beautiful and wonderful and amazing every single time. So I'm working with MarineGEO, and what MarineGEO is it's a program started by the Smithsonian that is trying to sort of do a global study of coastal biodiversity. And so what that means is that the Smithsonian, both through its own marine stations and by partnering with other universities and organizations, is setting up monitoring and experimentation that looks at the number of species, the types of species, how they persist, why they are or are not in particular areas, all over the world on a very long time scale. And that's kind of really unique. Ocean observing systems are sort of a common thing that you might find. And when I say that, what I mean is things like buoy systems that monitor temperature and salinity all around the world. These large networks, these are very sort of common things, but we've sort of taken that idea of the large marine network and translated it into a close coastal region and focused mostly on the biology of the fishes and the invertebrates that live there. And also the other really cool, unique thing about MarineGEO is we're thinking in very long time scales. So when I talk about, what MarineGEO will look like, we're thinking, 50 years from now. What sort of data will we accumulate to answer really important questions, since the world is changing at a faster rate than ever, and it's really important for us to know how and why it's changing.
And that's part of why this is such a great location, since you already have information from years and years, decades and decades of
Exactly.
research.
Yeah, I mean, since there's like 45 years of information from Carrie Bow already, that's a great place to start. And a lot of the other places that we're partnering with outside the Smithsonian as well have lots of, we call it legacy data. It's this data that they've already accumulated at a site, and what we're doing is we're building on top of that by using our own standardized sets of protocols. And that's really great that we're standardizing this across all the sites, because what it does is it allows us to compare sort of apples to apples, how things look at different places. You know, in the past, there have been all sorts of different studies of marine life in coastal regions, but everybody uses slightly different methods, or everybody counts different things. So what MarineGEO is doing is trying to standardize that across the board, so that when we say we see this number of species here and this number of species here, we can know that that information was collected in the exact same way.
Right. And so I think you have some things to show us about how you collect that standardized information.
Yes, I do. So some of the things that we're doing as part of the MarineGEO suite of monitoring and experimental protocols are what we call the feeding assays. So these are ways that we can get at how much predation or herbivory is happening in different environments. And right here I have what we call a squid pop. This is a
zoom in on this
zoom in on this, and that's kind of a
you can see the little
ratty piece of squid here. I actually have some better pieces of squid right here, because this one has already been kind of munched on. So something like this little like standardized disc.
That little guy right there in his hand.
That right there, yep. So that's a little piece of squid that we affix to the end of these stakes. We then put out 25 of them in a given habitat. So the habitats that we look at here in Carrie Bow are coral reef, mangrove, seagrass, and sandy sediment bottoms. So we'll put 25 of these out, we'll leave them for an hour and see how long it takes for the little bait at the end to be consumed by fishes. We'll also come back 24 hours later and see how much is gone. So on the reef, that happens really fast, and usually by the time we put all these out, they've already been eaten by fish, and that tells us that there's lots of predation and consumption by fishes on these reef. But someplace like a mangrove swamp, you might not necessarily see that much consumption. I could put these out and leave them for an hour and maybe see only 2 of them consumed after an hour. Though after 24 hours, we'll see a larger proportion of them consumed. So this is how we get at predation and sort of consumption of generalist predators. This is sort of the other side of that assay. This is called a weed pop. And so that was dealing with predators and opportunistic consumers, and this is dealing with herbivores, so critters that like to eat algae and other plants. And what we do is we put out 20 of these in the same sorts of environments, and we have 5 different types of, I'm not going to say algae because we only have 4 algae, and we have 1 seagrass, which is actually a plant, not an algae.
Right.
We put these out, and we're able to find out a couple of things. First of all, we're able to figure out how much herbivory is happening through the same method that we do the squid pops, but we're also able to say something about what sort of resources the fishes prefer to consume. So if we see that one particular species of algae off this rope is being eaten on all the ropes, we can say that fish really like that type of algae in this particular habitat. And something that we're doing this year, which is really cool, is we're pairing these 2 consumption assays with GoPro video. So we take these little weighted GoPros and we stick 10 of these out to actually watch the assays for the first hour of deployment. And we can go back later and look at the video, and besides now just knowing how much herbivory and predation and consumption is happening in these environments, we can actually tell you exactly how long it took for that item to be consumed and the identity of the consumer, which is really, really cool, and we're looking forward to digging into this year.
Yeah, it's been fun watching you deploy those. And I have a question from a viewer, Lisa, wondering what of the sites is your favorite to explore? Mangroves, coral reefs, or seagrass?
Gosh.
Tough question.
That's a really tough question. I mean, I kind of a cop-out, all of them, but I would say that 2 really stick out in my mind, and those are the coral forereef environments. So when you go out on the reef crest here, I mean, this is the 2nd largest barrier reef in the world, so it's very dramatic, very exciting, very dynamic environment, and there's just so many different species that you can see all at once, and so to me that's very exciting, and maybe catching a glimpse of a larger pelagic fish that you might not see in any of the other environments. The other one that I really enjoy and love are the mangrove forests, just because when I came into this job I knew absolutely nothing about mangroves. And one of our researchers at the Smithsonian, Candy Feller, she took me out and basically gave me a lesson in natural history of the mangroves. And I realized what an amazing sort of resource they are and how much life they really support. So also the mangroves.
All right. And there's one other thing that you're doing here besides squid pops and weed pops, which is called the Reef Life Survey. I know you've done it in a lot of different places, and can you tell us a bit about that?
Yeah, so Reef Life Survey is really cool because it's a partnership between MarineGEO and researchers at the University of Tasmania who started this Reef Life Survey method. And And so we're kind of joining our two networks together in a way to sort of double the bang for our buck. And what Reef Life Survey is it's a standardized protocol for visually surveying fish and invertebrates, mostly on reef systems, but we've also adapted it to do seagrass and mangroves as well. And it's really cool. They made this method that is very user-friendly. It's very quite easy to do with a little bit of training, and as such, they've been able to use it as a citizen science initiative.
So viewers at home, they can get in on this.
Yeah, if you go to the Reef Life Survey website, you can check it out. Same thing with the squid pops too. There's a video of how to make your own squid pops as well. But Reef Life Survey, I think they just hit their 3,000th survey, I think. And that's an enormous number of surveys that they've done in reefs all over the world. So we've sort of adopted their method, and by doing the Reef Life surveys here in Carrie Bow, or down in Panama where we have another station, or in Hawaii where we just were last month, we're not only able to sort of collect data in a standardized way about the fish community for our own purposes, but we're able to also contribute to another network that is building out this sort of global data set of fish and invertebrates. So it's really cool.
Yeah, and I know you also were just in British Columbia, and I see Hakai Institute on here, so shout out to
Yes,
them as well.
yes. Hi to everybody at Hakai. We did the Reef Life Survey at Hakai. They just participated in a MarineGEO BioBlitz activity, which is essentially a census of marine diversity. And it was kind of cool because we did Reef Life Survey up there, and we collected some of the first records of particular species for Reef Life Survey up there. We added quite a few new species to their species list, which was great.
Awesome. So last question for you. What's your favorite part of this job?
Scuba diving.
Scuba diving.
Yeah, I really love to dive, and this was sort of the perfect opportunity to really get face to face with a lot of these environments. Because I originally didn't work in the sciences, or marine or otherwise, for many years, and it was a fateful diving trip that really sort of got me excited about the marine environment. And if anyone out there has ever considered or thought about maybe learning to scuba dive, I would definitely suggest it, just because not only is it sort of exciting and fun and new, but it also can teach you something about this entire world that's right in your backyard that you might not know about.
Thank you so much, Ross.
Yeah.
Hope you have good results this week, and you get everything you need to done.
Yes, indeed.
And I had a question. We're gonna walk around. Say bye to Ross. We're gonna walk around. So I got a question. If this marine lab is part of a house where all of the researchers live. So yeah, this main house has a wet lab, which we just snuck down and got a peek of, and then a dry lab, which we're going to go talk to another researcher at. But you're kind of getting a glimpse. I don't want to walk too far away from internet, which is stationed a little bit behind me, but you can kind of see this just past that flag. There's a little kind of house. That's where all the scuba gear and the compressor for the scuba tanks is housed. And then there's a nice little, to the left of it is another little house where some researchers live. I think it can house 3. And then a couple people live in or are staying in this house that we are in right now. And you can see some of those doors to rooms. There's also a nice library where people can check out books and hang out and relax after a day of scuba diving. So we're gonna, just for those of you, if you hadn't heard me before, we're at Carrie Bow Cay, which is a Smithsonian marine station. I'm gonna walk through here and give you another, well, it's a little windy. It's a marine station for the Smithsonian that's 15 miles off the coast of Belize. It's a little less than an acre in size, and we're here talking to MarineGEO researchers that are actively working in the field. So we're going to go inside. This is the dry lab. So this is where lots of the work happens at night after people get back from dives. And we're going to talk to Maggie Johnson, who's here, and she is a fellow with Maria. Hi, Maggie.
Hi.
She's a fellow with MarineGEO, and she's also part-time, or she's split between MarineGEO and the Smithsonian Tropical Research Institute.
Yes, that's right.
STRI, which is in Panama.
Yes.
So, Maggie, I wanted to ask you what you're doing here. What do you study?
Yeah, that's great. Well, very broadly, my research interests are in studying coral reefs and how they're impacted by people. And the way that I'm doing that is trying to use some standardized methods, much like Ross explained to you guys a little bit earlier, to measure different properties of coral reefs in different places across the world. And one of the things, or one of the aspects of coral reefs that I'm really interested in is trying to understand how they're growing and staying healthy over time in different places that have different exposure to local human impacts or global human impacts. And this is really based on the concept or the fact that coral reefs, the ecosystem is actually built by our reef building corals. And corals are these really interesting and unique combination of animals and plants, and as they grow they secrete or create a limestone skeleton, and it's that limestone or calcium carbonate skeleton that creates the framework for the entire coral reef ecosystem. And that ecosystem supports tremendous biodiversity and serves a lot of different important roles for people. And so what I'm doing is using this standardized substrate, these PVC plastic tiles that I have pictured right here. It's a standardized settlement unit basically, and I call this a calcification accretion unit, or a CAL for short. And I put these little devices on the reef at different sites, different places. So right now we're here in Belize, and I have these tiles, or I've had them out on the reef for the last year at about 7 different locations. I also have these out in Panama, in Bocas del Toro, where I'm based most of the time, where I have been for the last year. So now, after a year, I come back, I collect these tiles, and I see what has grown on them. And what I'm interested in is looking at who is there that's actually contributing to reef growth or reef accretion, and that tells us a little bit about how the reef is actively growing, which is really important for the long-term persistence of a coral reef ecosystem. And so I have here some tiles that have been out, or were out on the reefs in Belize. On them. Yep, so here we have a few different examples. So in this little plastic tile sandwich that I have, this is an example of a tile that was on the top. Okay, so here what I've done is I've collected the tile, I've put it in a drying oven and dried it, and now I'm going to take this back to Panama with me and do some more analyses on it to determine who is here and how much they're growing and how much they are producing that limestone skeleton that I was telling you about. And so this stuff here is crustose coralline algae, which is another really important aspect of reef growth. And it's one that is a little bit less studied compared to the corals. So we've got our crustose coralline algae, and then this shows you the inside of the tile, which is this cryptic space in there.
So that space in between there?
Yeah, so there are a lot of critters that like to live in cracks and crevices because they're protected from things that might want to eat them. And so when we open up our tile after a year, we see all kinds of interesting things in there. Like we've got a sponge here that's dried out, we've got a little scallop shell. And so I've dried all of these, taken back to Panama, and by looking at these tiles and comparing them from different reefs across the world and across the Caribbean, it tells us a little bit more about how reefs are growing and how they're impacted by different human activities.
Great. So you mentioned kind of why it's important for these reef building organisms in the ecosystem, but how, for the people who are at home sitting at a desk, like how does that connect to them? Why is it important to them?
Right. So our coral reefs serve a lot of different, or provide a lot of different services to people. One that's related to this ability of the reef to, or the ability of this reef to grow and to accrete, is the physical protection that a reef provides to a coastal shoreline. So the physical barrier of a coral reef protects coastal lands from damaging wave and storm activity. Right? So having that physical barrier is really important for protecting those coastal areas from potentially damaging activity. And then in addition, our coral reefs are also really important for supporting a variety of different fishery species, and also for economic revenue from things like tourism.
I have a question here. How did you determine the material you use for your tile? So it's
Yes.
PVC plate.
PVC plate, so that's a great question. So we decided to go with this PVC for a couple of different reasons. One is that it is relatively affordable and is something that I can build myself, which is key for keeping costs down. Another one is that, so what I've actually done is roughed up the surface of this PVC. Normally the PVC tile is, or plastic is pretty shiny, and so I've roughed that up. And we found that coralline algae and other reef organisms really like to settle or to grow on bottoms that look like this. And we know this from putting out other instruments that are made of plastic, and you have all kinds of fouling on those instruments. And so we use these PVC plastic tiles because we know things like to grow on them. They're relatively lightweight, so it's good for traveling around with a box full of plastic tiles. And again, it's relatively affordable and easy to recreate, which means that we can use this as a tool to implement all across the world. It's really easy to use.
Great. So have you seen, you have these at a couple of different sites already.
Yeah.
Can you like see any changes between, over time or between sites yet, or is that still to come?
That's still to come. So I'm just finishing up the first year of my postdoctoral research with the Smithsonian, and at the start of that year, I deployed these calcification accretion units in Panama. We have deployed them here in Belize. And so just now I'm collecting the tiles to see what has happened over the last year, and it's the start of what could be a monitoring program that we can look at over time and see how things are changing. But what I can tell you is that the reefs of Belize and what's growing on these tiles in Belize is very different from what we see in other parts of the Caribbean where I work, in Bocas del Toro, Panama. For example, we have lots of coralline algae and reef-building algae growing on some of these tiles from the forereef here in Belize. And in Bocas, and some of the sites that are more impacted by people from things like nutrient pollution, sewage and land runoff, we see a lot more fleshy organisms growing that aren't contributing to reef growth. And so that's kind of what we're trying to get at, is what environmental conditions create these different reef communities.
One question that popped up, which is I think related, was about if certain species preferentially like this tile substrate. Do you know if there's, it sounds like it's really just based on ecosystems.
Yeah. So so settlement tiles have been used, this type of settlement tile has been used for a long time by researchers, right? So some people use ceramic tiles, some people use carbonate tiles. I couldn't tell you exactly. I know that corallines, crustless coralline algae, which is one of the groups of organisms that I'm really interested in, do prefer to settle on this PVC, which is useful for the research that I'm doing. There are some interesting studies that look at specific differences between carbonate tiles, ceramic tiles, and things like that. So there's some more
have to make a
information.
choice.
Yeah, you have to make a choice and then stay consistent over time. Yeah.
And finally, what's your favorite part about this job and the work that you do?
Yeah, so the obvious answer is the scuba diving and traveling and being able to go to some really amazing reefs and amazing islands. So that's one, but another part that I like about this job is that I really feel like I'm giving back to the reef ecosystems, and that I'm hopefully making a difference by doing this research in what's going to happen with our coral reefs into the future. So there's a little bit more motivation to keep going when the work gets a little tough, because we spend a week here on one of these beautiful islands doing scuba diving, and then we spend the rest of the year in the lab and at the computer analyzing the data and running the samples and trying to publish the results. So that keeps going through some of those long times.
And we got another quick question. You talked about some of the groups you're looking at on those tiles, which are corals and other invertebrates. A question about if you're looking at the bacteria, and also if you are using only morphological taxonomy, or are you just...
Yeah, that's a great question.
How are you looking at those?
I'm not looking at the bacterial or microbial community, but I know that there is a new MarineGEO postdoc who is, I believe, studying the microbial communities and settlement plates, so that's something coming down the line from MarineGEO. And then the other question was...
how do you morphological?
Yeah, morphological. I'm not doing genetic assessments. I'm interested in really broad patterns, functional group, taxonomic groups. So I'm not doing specific genetic testing of these different things. And so I'll analyze them, these dried samples, and then also through photographic analysis.
For those who might not know what morphological mean, can you kind of give us a sense of what you're talking about?
Yeah, so it's basically deciding what species you're looking at by how something looks, right? So the physical traits, physical characteristics.
Instead of those genetics.
Instead of the genetics.
Got it. All right. If you have other questions, we're going to say bye to Maggie for now. And I know that there are other questions coming in, and we are going to respond to those later via comments. We really want to try and get to all of them, but we're running out of time here. People have to go back to the field and get some dives in. So please do, you know, share this with your friends and your coworkers, your family, and please add questions as you think of them. This will be posted, the video, on the Ocean Portal Facebook page, and we're really glad that everybody could join us here on Carrie Bow Cay.